Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I will be going over stages of catabolism of amino group nitrogen. So as the title of this lecture says, we are going to go into how exactly amino group nitrogen, especially the alpha carbon bound amino group nitrogen, how that will be separated from an amino acid and how it is transferred from an amino acid or from the peripheral tissues all the way to the liver for urea formation. Let's jump into the details. Now the catabolism of amino group nitrogen can be studied in 4 stages. One is transamination reaction, where there will be transfer of alpha amino group from an amino acid to alpha keto acid to convert that and an amino acid. I'm going to explain that to you in the subsequent slides. And then the second stage is oxidative deamination, and the third stage is ammonia transport, and the final stage is urea formation in the liver. Let's get into our first stage. It is the transamination process. All amino acids undergoes transamination with the exception to lysine, threonine, glycine, proline and histidine. With the exception to these 5 amino acids, all other amino acids will undergo a transamination reaction. Reaction is very similar, general reaction that I am going to explain. Lysine, threonine, glycine, proline and histidine will undergo a direct deamination process. Now the transamination reactions are reversible. That's the first thing you should remember. After remembering how many amino acids out of 20 standard amino acids undergo transamination reaction, which is 15 of them, what are the exceptions? Exceptions you should remember: lysine, threonine, glycine, proline and histidine. And then the second point is all transamination reactions are reversible reactions conducted by a specific transaminase enzymes, one for each one of the amino acids. Like if we are looking at alanine transamination, we have alanine transaminase enzyme. If you are taking aspartate transamination, we have aspartate transaminase enzyme. If you are considering tyrosine transamination, we do have tyrosine transaminase enzyme, like that. And one thing that is common for all of these transaminases, or every transaminase, they need a coenzyme which is an active form of vitamin B6 called pyridoxine, and the coenzyme here is a pyridoxal phosphate. Pyridoxal phosphate, it acts as an intermediate for a transfer of amino group from an amino acid to an alpha keto acid to convert that alpha keto acid into an amino acid. Here you are seeing a general reaction of transamination where an amino acid is written and which has got an amino group. We are interested in the amino group, alpha carbon amino group. Make sure you pay attention to that, focus on that alpha carbon amino group which I have highlighted now. And also in the transamination reaction, the requirement is you need an amino acid and you need a keto acid. These are the 2 requirements. You should have an amino acid, you should have a keto acid, because the alpha keto acid, it removes an amino group from the alpha amino acid. So the amino acid is going to act as a amino group donor and alpha keto acid is going to act as an amino group acceptor. So that means you should have both. So in the transamination reaction, an amino group donor is always an amino acid and the amino group acceptor is always a keto acid. So amino group will be transferred from an amino acid initially to pyridoxal phosphate. I'm going to write PLP there with amino group transferred, and from there amino group will be given to alpha keto acid, because alpha keto acid is an acceptor of amino group mediated by the PLP, pyridoxal phosphate, which make a shift bond with the amino group there. Now once alpha keto acid receives that amino group from PLP, and that alpha keto acid is now converted to a new amino acid, while your original amino acid, it will be converted to alpha keto acid because it has lost its amino group in its alpha carbon. And this is how a transfer of amino group happens. Now the new amino group is present here in the new amino acid. I am going to highlight that with the green color. So this is how a amino group is transferred from an original amino acid to an acceptor alpha keto acid mediated by pyridoxal phosphate. Thereby, an alpha keto acid will be converted to a new amino acid, while your original amino acid will be converted to a alpha keto acid. So that's how the transamination reaction works. Now generally in a transamination reaction, an amino group is transferred from an amino acid to an alpha ketoglutarate. Now we are getting into the specifics. So that means in every transamination reaction, alpha ketoglutarate, it is an acceptor of an amino group. And when an alpha ketoglutarate accepts an amino group, and that alpha ketoglutarate will be converted to glutamate. So when a transamination is going on from different amino acids, always a glutamate is the new amino acid that is made. Make sure you remember that. Okay, so in this process, pyridoxal phosphate, which mediates the transfer of amino group, will have that ping pong mechanism. That means it will jump from one state to another state as the transamination is going on. That is shown in this general figure down here. As you see, an amino acid which carries an amino group will be first, it's going to give its amino group to pyridoxal phosphate, which in turn becomes pyridoxamine phosphate. Look at the amine group there, pyridoxamine phosphate. So I'm going to highlight that amine group, pyridoxamine. So amino group from an amino acid given to pyridoxal phosphate to become pyridoxamine phosphate, and now that pyridoxamine phosphate is going to give the amino group to alpha ketoglutarate. Alpha ketoglutarate is always an acceptor of amino group there from amino acids, and that alpha ketoglutarate, once it accepts that amino group, and it will be converted into glutamate. That means amino acids amino group via pyridoxal phosphate is now present in glutamate, and the glutamate is the new amino acid that is made in this transamination reaction. Like this, amino acids undergo transamination, 15 of them undergoing transamination to make a glutamate. So that means a common amino acid that is made in the transamination reaction is glutamate. So it is easier to handle a single amino acid glutamate to take that into the liver for deamination process, and then put that amino group into urea cycle so that urea is thrown out of our body. Instead of having multiple amino group carrying amino acids, it is easier to handle one amino acid which is bringing amino group from all the other amino acids and transport it to the liver. It's kind of easier to handle a single amino acid here, and that single amino acid in the transamination reaction generally which carries amino group from most of the other amino acids from the peripheral tissues is glutamate. Remember that. The reason for that is alpha ketoglutarate always accepts a amino group from other amino acids. Although these transamination reactions are reversible, but in the peripheral tissues, they are all going from amino acid to pyridoxal phosphate to alpha ketoglutarate, and alpha ketoglutarate to glutamate, and glutamate is transported. although glutamate can undergo further modification before transporting into liver. So here is a little more details on the transamination reaction for your better understanding. Now we are taking a specific amino acid instead of just saying it as a general amino acid 1 out of the 15 and all that. So here we are taking an example of aspartate to explain transamination reaction. Pay attention to the bidirectional arrows here because transamination reaction is a reversible reaction. It can run in either direction depending on the gradient. Pyridoxal phosphate is the common thing. I already explained you the role of pyridoxal phosphate. Now our amino acid interest is aspartate. So I'm going to color or highlight amino group of aspartate. Let's track that. So the amino group of aspartate undergoing transamination reaction. I told you always the acceptor of amino group during transamination reaction is α-ketoglutarate, which is a TCA cycle intermediate. By accepting the amino group, α-ketoglutarate will be converted to glutamate, and now your amino group is present in the glutamate. That means the amino group that was there in the aspartate is now present in a glutamate. That's why I have colored it, highlighted it same color. Now what happened to the aspartate then? See, the aspartate carbons will be present in oxaloacetate now. So for that I am going to show you with the different colors so that you can follow where exactly the carbons goes. So I am going to color aspartate carbons with the cyan color here. Okay. So at the end of transamination reaction, oxaloacetate carbons are same color as that of aspartate carbons. That means the carbons of oxaloacetates are actually carbons of aspartate, whereas the amino group of glutamate is actually the amino group of aspartate. Same color there. Now let's look at what happened to the carbons of α-ketoglutarate. I'm going to color that now with a different color. So α-ketoglutarate, I'm going to color this with the violet color here, and you can expect or guess that α-ketoglutarate carbons are found in a glutamate. This is the glutamate. Glutamate carbons are actually the α-ketoglutarate carbons, while the amino group of glutamate is coming from aspartate. So like this also you can track the carbons and also the nitrogen. Because why you need to do this? The reason for that is, if I am radio labeling certain groups in an amino acid and asking a question where exactly that particular group is found at the end of transamination. So if I am labeling a nitrogen atom in the aspartate, so I am going to color it red. That is the radio label. Think that it's a radio label. My question is, at the end of transamination, where do you find the radio labeled nitrogen after the reaction catalyzed by aspartate transaminase enzyme, AST, because the reaction is catalyzed by AST, aspartate transaminase. So where do you find at the end of transamination, where do you find the radio labeled nitrogen? So we are going to find that in the amino group of glutamate. That's how we can radio label. On the other way, let's radio label carbons of oxaloacetate. I'm going to radio label all the carbons present in oxaloacetate. And my question is, at the end of transamination reaction, where do you find the radio labeled carbons of oxaloacetate? We're going to find them in aspartate, because aspartate carbons are all actually coming from oxaloacetate, because we are running the transamination reaction in an opposite direction in making aspartate from oxaloacetate using amino group of glutamate, and the glutamate carbons will be actually the carbons of α-ketoglutarate. So like this, a radio labeling question can be asked to test the concept whether or not you have understood the concept. Okay. I hope you got it. Now once we get glutamate as the common amino acid which has received amino group from all other amino acids which are undergoing transamination reaction. So the amino group collected by transamination reaction as glutamate, and the glutamate is funneled into oxidative deamination reaction to release its ammonia in the liver. That's why I was saying it is easy to handle one amino acid rather than 17 amino acids. Okay, so ammonia which is released during oxidative deamination of glutamate will be funneled into urea cycle in the liver itself. Because the oxidative deamination of glutamate, which is conducted by mitochondrial glutamate dehydrogenase enzyme, releasing ammonium ion, and the ammonium ion going into urea cycle, and first two reactions of urea cycle are also there in the mitochondrial matrix. So it makes it easier for handling ammonium ion in the mitochondrial matrix. So look at the compartmentalization. Glutamate dehydrogenase is there in the mitochondrial matrix. Ammonium ion handling urea cycle first two reactions are also there in the mitochondrial matrix. So that is how it is compartmentalized in the liver. Now let's look at the glutamate dehydrogenase catalyzed reaction. See, the glutamate which is coming from transamination of majority of other amino acids is undergoing glutamate dehydrogenase reaction, where the amino group, alpha carbon amino group, is directly removed and released as ammonium ion, and the rest of the molecule is released as α-ketoglutarate. And this α-ketoglutarate, it can undergo further transamination reaction, or simply it can go into TCA cycle and continue the TCA cycle. Now the thing is, glutamate dehydrogenase is a special enzyme because glutamate dehydrogenase, it has got an ability to use either NADs, which will be converted into NADHH+, because it's a catabolic reaction. Glutamate is just undergoing deamination and energy is captured in the form of, basically there is a reduction of NADs into NADH+, H+, or glutamate dehydrogenase can use NADPs, and NADPs can be converted to NADPH plus H+. But the thing is, since glutamate dehydrogenase reaction is in a bidirectional reaction, it's a reversible reaction. But note that whenever, from left to right reaction, going from glutamate to alpha-ketoglutarate and release of ammonium ion, this is the predominant type of reaction going on in the liver. This is the direction of reaction that is going on in the liver majority of the time, predominantly. Reading it from right to left, that is fixation of ammonium ion to alpha-ketoglutarate's carbon skeleton to make glutamate. During that time, NADPH is getting into the reaction and released as NADP+. That is the one which is preferred there. So while it goes from alpha-ketoglutarate to glutamate, done by glutamate dehydrogenase, which is ammonium ion fixing, so NADPH is preferred. When glutamate is undergoing deamination, NAD is preferred. Just have an idea on that. Important, just in case. Okay, so that is what is a unique role of glutamate dehydrogenase enzyme. And also note that the glutamate dehydrogenase enzyme, it is regulated by nucleotides depending on the energy status of the cell. If there are more ADPs and GDPs, more ADP and GDP means lack of energy. So lack of energy means cellular breakdown is going on. Cellular breakdown means even protein breakdown can go on, releasing more and more amino acids and more transamination reaction. More and more transamination reaction means increase in the levels of glutamate under fasting state, catabolic condition. You know, catabolic condition. During catabolic condition, there will be more increase in glutamate, and that glutamate has to be undergoing glutamate dehydrogenase reaction to make ammonium ion released and α-ketoglutarate. So ammonium ion will be handled by the urea cycle in the liver. So under catabolic conditions, you expect there is increase in urea levels because there will be increase in the activity of glutamate dehydrogenase enzyme. Remember that. Now let's look at the opposite condition. When the cellular levels of ATPs and GTPs are more ATP and GTP. More ATP and GTP means it is a higher energy status, like a fed condition. If the person is in fed state, fed condition, and basically stating that there is no catabolism going on, at that time what will happen? So this ATPs and GTPs, they will have a inhibitory effect on glutamate dehydrogenase enzyme, and that means there will be decrease in glutamate and decrease in glutamate dehydrogenase reaction. Overall, there will be not a significant increase in urea formation unless there is a person has taken too much protein diet. That is a different story. So like this, a glutamate, handling of glutamate is important in the liver, and only the glutamate which is coming from all the other transamination reaction, it is easy to handle glutamate by fuel, funneling that into oxidative deamination reaction, releasing ammonium ion and funneling that ammonium ion into urea cycle, so that urea is made in the liver and thrown out of our body in the urine by the kidney. So all the points that I was saying in the previous slide, just to recap the points from the previous slide, here is a mitochondrial glutamate dehydrogenase enzyme catalyzing the reversible reaction, where it is the only human enzyme in the body that can use both NADs and NADPs as coenzymes. Only thing is, we'll have to look at the direction of the reaction. Going from breakdown of glutamate into α-ketoglutarate and releasing ammonium ion, NAD is preferred at that time. Going from fixation of ammonium ion to α-ketoglutarate, during that time NADP is preferred. Make sure you remember that. So that's what I have written here. NAD is used in the oxidative deamination of glutamate, whereas NADPH is used in the ammonium fixation, which generally happens in the perisinusoidal areas of the hepatocytes, where ammonium ion is more because it is coming from the portal circulation. So glutamate dehydrogenase is allosterically activated by ADP and GDP, indicating lack of energy status, catabolic conditions, where more and more glutamate might come into the liver. And allosterically it is inhibited by ATP and GTP, indicating a high energy status, where proteolysis might be relatively less. So these are some of the points that you must remember with handling of glutamate. Now, we have seen one of the enzyme that has got an ability to fix ammonium ion. So the slide here, it is showing you 3 enzymes that can fix ammonium ion in our body. One of the enzyme that we have already seen in the previous slide, and that is glutamate dehydrogenase, fixing ammonium ion to α-ketoglutarate. Usually this happens in the perisinusoidal regions of the hepatocytes because of higher amount of ammonium ion coming from the intestine in the portal blood. So in the figure here, that reaction is shown where α-ketoglutarate carbons accepting ammonium ion and becoming glutamate, where NADPH is used as a coenzyme, and the reaction is catalyzed by glutamate dehydrogenase enzyme. Ammonium ion is fixed to α-ketoglutarate. Now once you get glutamate there, this can happen in the muscle, peripheral tissues and all. Now the glutamate can accept ammonium ion to its side chain as amidation process to make a glutamine. So there is an amidation of glutamate to convert that into glutamine, and this is done by glutamine synthetase enzyme that uses ATP. As the name says, synthetases uses ATPs to condense, to fix. So that's the second enzyme which will fix ammonium ion to an organic molecule. That means a carbon skeleton of α-ketoglutarate, it has got an ability to fix 2 ammonium ion. Alpha-ketoglutarate has got an ability to fix 2 ammonium ion to make glutamine, and the glutamine can get out of the peripheral tissues into the blood, and it can go to the liver, and glutamine can release those 2 ammonium ion for the urea cycle. That's how ammonium ions can be carried from the peripheral tissues all the way to the liver. So remember, if you see 1 glutamine in the blood, that means there are 2 ammonium ions in that. In fact, the glutamates and glutamine levels in the blood are, when you compare compared to amino acids, total amino acids in the blood, available amino acids in the blood, glutamate and glutamine itself, they represent around 40 to 50% of the amino acids that you are going to find in the blood, because every tissue which is sending this glutamate and glutamine out of their cells into the blood, so that they will be going to the liver, because transamination reaction will make glutamate, and glutamate can be further undergo amidation to become glutamine. So glutamate and glutamines are the predominant types of amino acids in our blood. Remember that point, very important. Now, what is the other enzyme that is involved in the ammonium ion fixation? It is present, it is the very first enzyme in the urea cycle, again present in the matrix of mitochondria. And also remember these 2 enzymes here, that is glutamate dehydrogenase, glutamine synthetases are mitochondrial enzymes. Likewise, carbamyl phosphate synthetase 1 is also a mitochondrial enzyme. So what it does, it is going to fix ammonium ion to bicarbonate. So where carbamyl phosphate synthetase 1 enzyme, where ATP is converted into AMP and the product that is made is carbamyl phosphate. This is the very first reaction in urea cycle that we are going to see. So with that there are 3 enzymes which can fix ammonium ion to an organic molecule, that is glutamate dehydrogenase, glutamine synthetase and carbamyl phosphate synthetase 1 enzyme. Now, other than glutamate dehydrogenase reaction, which is involved in deamination of glutamate, there are other alternate pathway for deamination, and that will be done by amino acid oxidases. So there are 2 kinds of amino acid oxidases. One is L-amino acid oxidase, and L-amino acid oxidase acts on all amino acids except hydroxyl containing amino acids and dicarboxylic amino acids. What all the hydroxy amino acids and dicarboxylic amino acids? Hydroxy amino acids, we have 3 of them. One is serine, other is threonine, and the third is tyrosine. Dicarboxylic amino acids, basically meaning these are acidic amino acids having 2 carboxylate group, and that is aspartate and glutamate. So other than these 5 amino acids, all other amino acids can be handled by L-amino acid oxidase and they can be deaminated. Similarly, and this L-amino acid oxidase is generally present in the kidney. So there are D-amino acid oxidases which function during the degradation of D-amino acids which are of bacterial origin from our intestine. Now both these type of L-amino acid oxidases, they are present in the peroxisomes of the liver, and also you're going to find them in the kidney. Now, what is the general deamination format here for amino acid oxidases? Now, the amino acid here containing alpha amino group catalyzed by amino acid oxidase, which will prefer FMN or FAD which will be reduced into FMNH2 or FADH2. And this is the, as you know, FADH2 can go into electron transport chain second complex to give energy. That's how the amino acid breakdown, basically amino acid catabolism here can give some energy. And also the carbon skeleton of amino acid after removal of the amino group, alpha carbon amino group as ammonium ion. So it will be getting out as alpha keto acid, which can also be used for energy purpose, or it can go into any other metabolic fate in the cell. Now, one thing that you need to remember here is not only about ammonium ion that is released. Here it is shown as ammonia. I'm going to come to that point a little later. So the point that I would like to bring here is there is a release of hydrogen peroxide. That means whenever amino acid oxidases are acting on the amino acid for deamination process, there will be release of hydrogen peroxide, and that's why you see this type of enzymes are located in the peroxisomes. Why? Because peroxisomes, they are rich in catalase enzyme. There is highest concentration of catalase in the peroxisomes, where hydrogen peroxide is neutralized into water and an atom of oxygen, which will be released as a molecular oxygen. And also it can be a source of reactive oxygen species. So overall, the catalase enzyme is going to neutralize hydrogen peroxide in the peroxisome. Now coming to ammonium ion that is released here. See, this ammonium, that is NH3 is ammonia. You need to have idea on this. This is ammonia. Ammonia is a gaseous molecule, volatile, whereas ammonium ion NH4 plus is an ionized form of ammonia. And in our body, for every 1 molecule of ammonia, there will be 100 molecule of ammonium ion because of the pH-pK relationship. So the pK of ammonium ion is around 9.5 and the pH is 7.4. The pH is 7.4 and pK is 9.5. So that means pH is less than pKa. So when a pH is less than pKa of the group, so group will undergo protonation. So molecule exists predominantly in its protonated form. That means ammonia will be protonated to become ammonium ion. That's why majority of the ammonia in the transamination reaction will be present as ammonium ion. Only when ammonium ion has to go out and go into the cell, it will be present as ammonia NH3 plus. Other than that, it will be existing as ammonium ion. Remember that. So in the blood, for every 1 ammonia that you see, you are going to see 100 ammonium ion. Very important point. You got to remember this. Now let's move on to see what are the other sources of ammonium ion in our body. Sources of ammonium ion is given in this particular figure. Carefully go over the contents. Some of them are really important. One is a purine nucleotide cycle. Whenever purine nucleotides are undergoing a degradation, so part of that degradation pathway will give ammonium ion as the source. Pyrimidine nucleotide degradation also releases ammonium ion. And then breakdown of glutamine into glutamate, that amide nitrogen can be released. Breakdown of glutamate into α-ketoglutarate by glutamate dehydrogenase reaction. Breakdown of serine into pyruvate. Breakdown of threonine into α-ketoglutarate. And histidine conversion into 𝐒𝐫𝐞 and asparagine conversion into aspartate. These are some of the sources of ammonium ion. But one thing that I would like to bring to your attention here, and the most important thing, and it has got a clinical application, and that is one of the significant source of ammonium ion in our blood is the one that is coming from portal circulation. So I am going to color that differently here. So here this is the one that contributes significantly to the ammonium ion that is coming into the liver through portal circulation. Now what happens here in the intestine? So the amino acids in the intestine coming from the diet, protein digestion into amino acids, and these amino acids in the intestinal lumen will be acted on by the bacteria, some of them. So the bacteria going to convert amino acids, release their amino groups into ammonium ion, and this ammonium ion, specially released as ammonia, enters into the enterocytes and from there enters into portal circulation and contributes significantly the ammonium ion that is present in the portal blood. Even the urea can undergo uricase reaction by the bacterial action, releasing ammonium ion and contribute to ammonium ion in the portal blood, which will go to the liver. So my point here is the intestinal bacteria are the significant source of ammonium ion, which is entering into the portal blood. Ultimately, that ammonium ion has to be handled by the kidney. Why? Because as the portal blood enters into the into the liver, so portal circulation is very sluggish. So slow blood is flowing slowly. So the contents in the portal blood is taken up by the hepatocytes because there is sufficient time to do that. And one of the content that is taken out of the portal blood significantly by the hepatocytes is the ammonium ion, which is entering into the hepatocyte as ammonia. So as it is taken in, so the ammonium ion is diverted into urea cycle to convert that ammonium ion into urea. Because if the liver do not convert ammonium ion into urea, if the liver do not take up the ammonium ion from the portal circulation, that ammonium ion will eventually be getting into the systemic circulation, and then it will be having a systemic effect, especially the neurological effect, because ammonia toxicity will lead to severe neurological issues. In order to avoid all that, urea cycle has to go on in the liver, and liver has to function properly to handle that ammonium ion which is coming from the portal blood. So in the liver disorders like cirrhosis of liver in advanced stages, so liver fails to remove that ammonium ion from the portal blood and the urea cycle fails, thereby excess ammonium ion will be elevated in the blood, and that will lead to neurological issues, ultimately giving rise to something called as hepatic encephalopathy. And one of the way to reduce hepatic encephalopathy is to reduce ammonium ion levels in the blood coming from the portal circulation. And these broad spectrum antibiotics, they are going to kill this intestinal bacteria by sterilizing the intestine, thereby decreasing the ammonium ion that is entering into the portal blood. And that's where this part of the figure makes it a high yield or important. Now let's move on to see what is the role of glutamate, which is one of the amino acid that is coming out of transamination reaction. And this glutamate, it can be helpful in the formation of a new amino acid, because transamination reactions are reversible reaction. Glutamate can donate its primary amino group, that is NH3 plus, to a new alpha-keto acid. And once a alpha-keto acid accepts that amino group coming from glutamate, an alpha-keto acid becomes an amino acid, and the amino group is now present in this, and glutamate carbons will be released as α-ketoglutarate. Like this, a new amino acid can be made using amino group nitrogen of glutamate. Glutamate can also be participating in the degradation of amino acids, especially glutamate carrying the amino group nitrogen from different amino acids will divert and funnel that amino group to urea cycle. How that is done? So the amino acid giving their amino group, I am going to write NH3 there as on the amino acids, undergoing transamination reaction. Alpha-ketoglutarate is an acceptor of that amino group. So amino group is now present in the glutamate at the end of transamination reaction. Now this glutamate, it has got two fate to go into urea cycle. One fate of that glutamate is to go into glutamate dehydrogenase reaction to give that ammonium ion, because glutamate dehydrogenase releases ammonium ion from a glutamate. That's a deamination, glutamate deamination process by glutamate dehydrogenase enzyme. So ammonium ion can go into urea cycle. Other fate is glutamate undergoing transamination reaction done by aspartate transaminase enzyme and giving that its amino group to aspartate, and the aspartate has the amino group now. So that means glutamate, it will undergo two fate. One is to go into glutamate dehydrogenase reaction, other is to go into transamination reaction. So giving aspartate and ammonium ion, both of them will enter into urea cycle to make urea. Note that urea is NH2CONH2. In that NH2CONH2, which is the formula for urea, one of the nitrogen will come from aspartate, other nitrogen will come from ammonium ion. So that means in a urea molecule, glutamate's nitrogen, meaning two glutamates, one glutamate going into glutamate dehydrogenase reaction, releasing ammonium ion, and that ammonium ion going into urea cycle, contributing to one of the nitrogen in the urea that is made, and glutamate going into another glutamate going into aspartate transamination reaction, making aspartate and giving the second nitrogen of urea. So that's how urea is made, by using two nitrogens coming from two glutamate, and those two glutamates are coming from transamination of all other amino acids. That's how amino group nitrogen from all the amino acids, they will be transported to glutamate by transamination reaction, and the glutamate ultimately transfer those amino groups either to release that as ammonium ion by glutamate dehydrogenase reaction in the mitochondrial matrix, or make aspartate. So both of them are needed for urea cycle, and urea is made, which is NH2CONH2. So this is how amino group is transferred from one amino acid to another amino acid, and that amino acid is glutamate, and glutamate has a two fate which will transfer those amino groups to urea cycle. These are the stages of catabolism of amino group nitrogen. I hope this detailed video has helped you in understanding this very important and seemingly a difficult topic. Thank you.